carbon, just copy down one of these nucleotides. See how this one's upside down? Look, there's the 5, there's the 3. So if I'm going to add here, I'm going to go this way. 5 to 3. Okay? Always in that direction, like left to right. 5 to 3. Okay? All right, here we go. Prokaryotes and eukaryotes, again. And we're never going to stop with this prokaryote versus eukaryote stuff. Okay? We're eukaryotes, they're prokaryotes. They want something from us, they're making us sick. Right? We want to be healthy, we got to know everything about the distinction between those two. All right, prokaryotic ribosomes, chromosomes, they're not the same. Not the same as ours. Okay, first of all, when we're talking about prokaryotes, the chromosomes are few in number. That makes sense, they're incredibly small. Okay? But also, guys, the chromosomes are circular. We're linear. You guys draw chromosomes with an X, right? That's 2 lines. These guys do circles. And we don't have a nucleus. We have a nucleoid, which again is just a DNA spot. A place where DNA exists, not a structure where it's held, just a place in the cytoplasm. Okay? Circular, 1 to 2 max. Okay? And prokaryotic cells are also haploid. And let's really get away from our intro genetics and talk about what haploid really means. Haploid, hap is half. Half of 2. Okay, we have 2. Half of 2 is 1. A prokaryotic cell, everybody, only has 1 chromosome. That means 1 copy of a chromosome. There's not a mom copy. There's not a dad copy. There's just a copy. That's haploid. We're diploid. Mom had an egg with a chromosome in it. Dad had sperm with chromosome in it. His chromosome, her chromosome, hang out together. His chromosome 1 hangs out with mom's chromosome 1. Homologous, 1 and 1, 12 and 12, 15 and 15, 18 and 18, 22 and 22, right? There's 23 of ours. These guys do not have a mom and dad version. They just have 1. Okay? Because there's no mom and dad bacteria. Everybody get that? We're diploid because we got 2 parents. They don't have 2 parents. They got 1 parent. We double. Okay. Good. Gay. Don't care. Plasmids. So bacteria, guys, they've got chromosomes, and they also have these funky pieces of DNA called plasmids, and they're also circular, but they're very, 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 very small circles of DNA. Okay? Little circles, smaller than the circular chromosome, real small, and they only have a certain number of genes on them. And they're involved in a couple different things. I want you guys to know 2 of these 4. Okay? We got 4 different plasmids up here. The main role of plasmids is to make the bacteria, help the bacteria survive. Okay, this is the conjugation thing, right? We got the conjugation pilus, and we're going to give you some DNA. Most often when I'm giving you DNA, guys, I'm giving you a plasmid. All right? And the thing about it is, I've got to give you the fertility plasmid before I give you the resistance. The only one we really want to know is resistance. And before you can receive it, I got to give you a fertility plasmid. Okay? It's like, it's kind of like I got to give you some DNA, like first, for you to be able to receive the real DNA I'm about to give you. So I give you this fertility plasmid first, and that allows all these others to come inside, but I just want you guys to know the resistant factor, which is the most important of all. That when we discovered that bacteria are resistant to penicillin, it was a gene, everybody, on their plasmid that made the penicillin not work anymore. It made an enzyme that chopped up the penicillin. Okay? And so guess what it did? It started passing that plasmid to everybody in the colony. And then everybody that was receptive, everybody that had a fertility factor that was ready to go, got a little plasmid, and now they're resistant. And now we pass it around because everybody else that's not resistant dies, right? The ones that survive are the ones that receive the plasmid. It's a circle of DNA, a plasmid. For our purposes, a circle of DNA in bacteria only, whose job is to make you resistant to poisons. Okay? Job is to make you resistant, guys, to poisons. All right. This doesn't sound very good, but only 25 minutes left. All right. Okay. That's prokaryote. Circular, haploid, they got plasmids. Let's talk about eukaryotes for a second. Eukaryotes, chromosomes are in the nucleus. Right? We got a nucleus, chromosomes are in there. Y'all know that already. Unlike bacteria, everybody, we've got typically, right, the bigger you get especially, but, you know, bacteria typically one, eukaryotes typically more than one, typically way more than one, okay? Not circular, linear, as you guys know, right? Guys have a Y chromosome, you guys know that, right? We'll draw a Y. Those are lines that you draw, okay? Can't make a Y with a circle. Right, Dana? Is that funny? Just asking. All right, it's wearing off. No, not totally. Okay, eukaryotic cells don't have to be. But if you want to, hey, if you want to find diploid, there's only one place to look. Okay, that doesn't mean that everybody, guys, is diploid out here. You got single-celled amoeba, right? You know mom and dad amoeba. Those guys are haploid too. Okay. It's just that if you want to find diploid, the only place that's happening, guys, is eukaryotic cells. We're diploid, right? Anybody got a mom and dad? You know what I mean? You got enough room here, guys? Diploid. Quote, unquote, mom and dad. That's all that means. Mom's got a set. Dad's got a set. Come together with fertilization. I, as a zygote, have two sets. That's how it works. Okay. So, this is actually a big freaking deal. I didn't think it was a big deal, honestly, guys, until I started reading some more. Over the last couple weeks, I was off for 4 weeks, right? While the spring ended and summer started, there was 4 weeks in there. So I was, because I'm a geek, I was reading this book called Genes. And they have a whole chapter, in fact, they've got a couple chapters on this slide, on things called nucleosomes, guys, and this crap called chromatin, okay, and heterochromatin, and then chromosome. Historically, I would say, you guys don't need to know that. After reading the chapter, I don't want it, I'm still going to say it to you, okay, but I don't want to. Because this stuff is really, really, really important in genetics. Whether genes are on or off, whether you're going to have certain characteristics or not, oftentimes has to do if the DNA is packaged or not. Guys, how am I going to get to the DNA when it's all wrapped up like this? It's all packaged up. You can't do anything with that. You got to unwind it. You got to get naked DNA. This is not naked. This is DNA plus proteins called histones. Got to know it. Okay? All eukaryotic chromosomes are packaged the exact same way. They're all comprised of nucleosomes. And I take those nucleosomes and I wind them in specific ways, and the more winding I do, the different structures the genome or the DNA, the baby chromosome makes. There's essentially 2 stages on your way to full-blown chromosome. All right? 2 stages. I want you to know, guys, nucleosome, the things on this slide. Nucleosome, histone, and chromosome. Chromatin, heterochromatin, euchromatin, those are stages on the way to packaging, but let's honestly know what a chromosome is. It's a fully wrapped DNA molecule. Okay, guys, you guys know we have 23 different chromosomes, right? And then a mom set and a dad set, so 23 times 2 is 46. Okay? But what does that really mean? It means we've got 23 different piles of DNA. We got one pile, we wrap it up, it looks like chromosome 1. Another pile, wrap it up, looks like chromosome 2. 23 individual piles Dad's got a set of piles, mom's got a set of piles. We wrap them up and we call them chromosomes. Okay? All right. The beginning of forming a chromosome means you've got naked DNA that's wrapped, guys, around 8. It's an octet, 8 histones. And it's a wrap of 2. It matters. Again, reading those chapters over the summer really got it in to my skull when we talk about the details of the winding and the specific, there's names for these histones. They're colored the same. But there's H2 and H2 and H4 and H4, and there's this other one called H1 that's not being illustrated. I'm saying, in molecular genetics, it gets really crazy with the specifics. We're foregoing most of that. I just want you to know that we take a pile of DNA and we got to package it. Does anybody know why we got to package it? Mitosis. Why do we package? How in the hell are we going to move this pile of DNA? It's like a pile of yarn, guys, that could fill this entire building. But when we wind it properly, we can fit it inside this tub. That's the difference. If I want to pass the pile to somebody, isn't it easier to get it into a structure that fits in here and just do a shovel pass, right? This is where I'm counting on the guys here. Little gender communication. What's a shovel pass? What sport? Football. Okay. 40% of the NFL fan base is women. Okay? And you guys are like, duh. I know. Okay. Like, yeah. Have you ever watched it? Yeah, I get it. It's not because you know what a shovel pass is. I'm just saying. Okay. 8 proteins wrapped twice. That's what a nucleosome. Excuse me. That's what a nucleosome is. Okay? I don't care, guys, about linker DNA. Skip it. Nucleosome, that's the fun. You know how the atom, guys, is the fundamental piece of matter? Nucleosome is the fundamental piece of a chromosome. Okay? And a eukaryotic one specifically. Prokaryotes don't do this. They don't even have histones. Okay? So, I get these specific beads. This is called beads on a string. Don't matter, guys. They're just chunks called nucleosomes. And long story short, I take that pile that's got nucleosomes and I just wind the living crap out of it until I look like this. Okay? And we got some names for it along the way that comprise like multiple pages, small print at the graduate school level, getting nasty with really how these things behave and proteins and wacko stuff. Okay? But we don't really need to care about that. Nucleosome, histone, chromosome. Eukaryotic DNA packaging. I've got to get a ball of yarn the size that would fit this building into this container. That's a lot of winding, guys. Extremely tightly wound, a chromosome. Cool. Class dismissed. I need that. Okay? And not just for the South County crowd. Class dismissed for real. Okay? We'll pick it up, guys, tomorrow. That's part of the reason why class is dismissed. Okay? Because we got to do this again tomorrow.